this is the culprit for many disorders. But we will come to that. So this iron is delivered to our body from our daily diet. Our daily diet contains around 10 to 12, 20 milligram of iron, out of which only 1 to 2 milligram is absorbed in our body. This iron, this 1 to 2 milligram, which is absorbed in body, is utilized for different purposes like hemoglobin synthesis and other processes. And the rest is stored in body, mostly in liver. It can be stored in other organs also, but mostly the iron storage proteins are in liver. And this protein is termed as ferritin. So we must understand that there is no dedicated physiologic excretion mechanism for iron. So what we get, what we absorb, we utilize, and the rest is stored. Because of this, the absorption, the utilization, and storage must be highly regulated in human body. A steady balance of iron in our body. Otherwise, the amount of iron in human body can be less or more than our requirement. This tight regulation is termed as iron homeostasis. So we'll see a glimpse of how iron is absorbed in duodenum, in intestine. The food iron, that either heme or non-heme, reaches intestine, where the heme iron is directly absorbed via heme transporter protein. On the other hand, the non-heme iron is reduced to iron plus 2, which is then absorbed via divalent metal transporter. Actually, vitamin C, vitamin C is ascorbic acid, and that is an antioxidant. That means that is a reducing agent, plays a role in reducing the non-heme iron for further absorption. That is, the plus 3 iron is reduced to plus 2 iron, and that enhances absorption of iron. That is why you need citrus fruit for iron absorption. Now, once absorbed, the iron is transported across the blood serum via another protein called as ferroprotein. In blood, once the iron reaches to blood, the iron is transported by a protein called transferrin. Two atoms of iron are being absorbed by one molecule of transferrin. That is a dipheric transferrin kind of thing. So these dipheric transferrin takes the iron to different cells, organs, tissues, or whatever it is required. And there it is utilized further for different physiological processes. The excess iron, whatever is left after this utilization, is stored in liver, mainly in liver, using ferritin protein. So, if we see that after the iron reaches the cells in forms of dipheric transferrin, this dipheric transferrin, it is absorbed via transferrin receptor into the cells. So till now it was outside the cells in the serum. Now it is inside the cells using a transferrin receptor. So once inside the cell, the intracellular acidity increases. It is a natural phenomenon to increase the intracellular acidity, so that the transferrin leaves the bound iron in the cells, and this unbound transferrin, or the apotransferrin without iron, it goes back to the serum pool for another ferry of iron. So this iron inside the cells are then taken up by different organelle for further use. So we have seen how this are done and how it is being utilized. Now, after the iron reaches to cells in the form of dipheric transferrin, it is inside. Now we should know that how to regulate the absorption and the storage of iron. The homeostasis is actually carried out by a protein called hepcidin, which is released from liver. Now, once the body senses that the amount of iron which is absorbed by the body is more than that required, and the amount of iron that is being stored in liver is much more than expected, that is, body is receiving much more iron than expected, it secretes hepcidin. So what does this hepcidin does? This hepcidin protein binds to the ferroprotein. Ferroprotein was the protein which was responsible for taking the iron into the bloodstream. So it attaches this, the hepcidin actually binds to the ferroprotein responsible for iron absorption, and thus disables ferroprotein. So thus the absorption of iron is regulated. So in a summary, there is no excretion for iron, and iron homeostasis is thus absolutely necessary. Homeostasis will result in either iron deficiency, which create erythropoiesis, that is inefficient formation of hemoglobin, and that is detected by low hemoglobin level in your body. So that's why we call it anemia. And this hemoglobin deficiency can be solved by having iron-rich diet or iron supplement. But the problem is very bad, or this problem worsens when it is excess iron, which is termed as iron overload. But we must understand, why do we bother for iron overload? Because excess iron, whether it is detrimental or it is good for the body. So iron is a redox active metal, and it may switch between +2 and +3 states. The reaction inside the body which is responsible for the switching is named as Fenton reaction, where iron +2 get oxidized by intracellular peroxides to +3, and in this process it generates reactive oxygen species like hydroxy radical. This increases the oxidative stress, leading to detrimental processes like lipid peroxidation, DNA damage, and ultimately leading to cell death. This renders free iron, this term is very important, the free iron, very detrimental to human health. One thing, so we must remember that thing, that the Fenton reaction exists only for free or labile iron. If the iron is bound to some protein or something, so that becomes redox inactive. It does not take part in Fenton chemistry. So bound iron like dipheric transferrin, hemoglobin, and ferritin do not take part in Fenton reaction, and so these are harmless. So this is the beauty of nature, that the iron in normal cases are always bound or chelated to proteins or chelators. Otherwise, whatever excess iron we have in our body, which is free or labile, can take part in Fenton reaction. So, as we have seen, whatever iron is inside absorbed in the body is bound to ferroprotein to go into the bloodstream. Then it is bound to transferrin to be transported, and then it goes into the cells, and then it is utilized. So, where from this free iron comes in human body? This can happen only when the homeostasis is disturbed. That is, the absorption is iron is not properly regulated. As we have seen earlier, hepcidin regulates the absorption of iron. In certain hereditary conditions, the hepcidin is damaged, and so the absorption is not controlled, leading to uncontrolled absorption of iron much more than required. This eventually leads to excess iron in the plasma of the blood. If you see the natural progression of the disease, you can see this curve. Increased serum iron leads to increased liver iron, and ultimately to liver damage, and in extreme cases to organ failure and death. So when your iron is being absorbed in a higher amount, so where does it go? It goes into the bloodstream. After absorption, it goes into the bloodstream. It is bound to transfer. Now, body has a mechanism where it does not allow more than 30% transfer in saturation with iron in normal cases. However, the saturation level increases when excess iron reaches blood plasma. Eventually, when the transferrin capacity is exhausted, that is, all transferrins are bound to iron, iron in its free form appears in serum. So that is the non-transparent bound iron, or the free or labile iron. So that is redox active and is detrimental. So this is termed as labile plasma iron, or the non-transparent bound iron. This excess iron eventually reaches the organs and start depositing there, leading to iron-induced oxidative stress. As we have seen that it is very detrimental. So these non-transparent bound iron can be formed either by uncontrolled absorption in the hereditary hemochromatosis. We have seen that if hepcidin cannot regulate the iron absorption, so it can lead to excess iron in blood plasma, or in cases where a person gets blood transfusion. In cases of blood transfusion, you are having a, a excess of blood full of iron directly into the blood plasma. And each transfused blood contains approximately 200 to 250 milligrams of iron. This is the case for thalassemia patients, where they get frequent blood transfusions. Actually, 80% of the deaths in thalassemia cases are due to iron-induced toxicity, nothing else. So hence, all the patients who get frequent blood transfusion, in case of anemia, in case of thalassemia, etc., also are subjected to iron overload therapy simultaneously to get rid of excess iron. So in case of iron overload in blood plasma, what happens that it is enriched with excess dipheric transferrin as well as excess non-transferrin bound iron. The cells both are penetrated inside the cells. Transferrin bound iron through a transferrin receptor and the unbound iron are directly into the cells. So these excess iron is either taken up for further use or for storage. But the problem is that the traffic time between the iron penetration and the utilization, that is taking up this free iron for use, is in milliseconds, which is much more than the time scale of the Fenton reaction. So the labile or free iron, whatever we are talking about, gets enough time to participate in the Fenton reaction to produce oxidative stress inside the cells. This is very detrimental. The pool of intracellular labile iron is termed as labile iron pool. So the extracellular labile plasma iron leads to increase in intracellular iron, leading to the formation of labile iron pool. In fact, it should also be mentioned that when body senses more labile iron pool, that is intracellular iron, it senses that there is more intracellular free iron, and it is much more than the that is required or that is for utilization. It produces more and more storage proteins like ferritin, so that this excess iron can be stored accordingly. So if we look at the difference between the normal condition and the iron overload condition, we would understand that the non-transferrin bound iron, which was prevalent in the plasma, blood plasma, is responsible for the labile iron pool, that is the intracellular one. And this is the cause of many problems. Like, so as we have discussed, the reasons for iron overload can be either inherited, that is the hereditary reasons, like hereditary hemochromatosis, and this is one of the most important inherited reasons. There are other reasons, and all are associated with some dysfunction of the regulatory proteins. That is the secondary reasons are like long-term hemodialysis, frequent blood transfusions in thalassemia cases, alcoholic cirrhosis, where the liver gets damaged and iron is secreted from the ferritin, the storage protein. So this excess iron is mixed in the blood plasma. So these are the reasons of these are the reasons for iron overload. So the results are very toxic. Means as I have already told, the excess labile iron leads to oxidative stress, and that is manifested in human body by various diseases like cardiac disease, neurodegenerative disease, etc. One must remember that for every disease there are various reasons associated, and iron is only one of them. So it should not be thought that okay, iron is the only reason, so if I get rid of iron, Alzheimer's disease will be cured. No, not like that. But iron is one of the primary reasons for this Alzheimer's disease. So how do you diagnose an iron overload case? As I have shown you that in iron overload, so it can be detected primarily by detecting or measuring the labile iron in the blood serum. But that is not so easy because of many proteins associated. So the diagnosis relies on the other markers like transferrin saturation or ferritin levels in blood. As we have already seen, The transferrin saturation is higher in case of iron overload than in normal individual. Also, the natural body response increases the secretion of ferritin when it senses iron overload. So the doctors look for transferrin saturation level or the serum ferritin level for diagnosis of iron overload. If ferritin level is high, then it is iron overloaded. If transferrin saturation is higher than 10 or 15%, it is termed as iron overloaded. And there are direct methods also like doing liver biopsy or doing MRIs to detect iron deposition. But that is much more costly and involves some uh, sophisticated instruments and also time consuming. Now, the problem is of iron overload can be mitigated in several ways. The most primitive way is to suck the bad blood out of your body till the iron level is normal. This is a very primitive method, but it is followed in many regions till now. One of the major problem in this therapy is frequent hospitalization because you have to donate blood. And so it is having very poor patient compatibility, and it needs constant monitoring of the blood parameters, because when your blood parameters are abnormal, then you have to stop this phlebotomy. Another method, easier but less effective, is via diet control. The affected person has to take iron free diet for a long time till his or her serum iron level is normal, but it is not so effective. But the most promising and effective therapy is via chelation of the labile iron, and to take out the iron chelate out of the body via urine or feces. This is a kind of locking the bad guy in your lab and then throw it out of the system. So, as I have told you, that chelation therapy is the most effective therapy. So, there are many chelators for iron, as you know, and in the first session of this webinar, we we have seen some iron chelators also, some iron bound compounds. Those are all iron chelators. So, there are several chelators for iron. Starting from naturally available phytochemicals to the synthetic compounds designed to chelate iron. Among these, the siderophores are practically very important. So what are siderophores? Siderophores are natural uh, iron chelators which are found in which are found in algae or bacteria or something. So some lower organisms. So these siderophores are generally secreted by lower organisms, particularly for speciation of iron. So these are having lower toxicities, and the siderophores have been seen to be particularly useful for treatment of iron overload. There are many synthetic chelators also, like hydroxypyridinones or deferiprone, these kind of hydroxyquinolines, deferisirox. These are also having important role in chelation therapy. But among all of this lot, there have been only 3 approved chelators for clinical use. Despiroxamine, despiroxamine is a siderophore. So you know what siderophore is. Now, despiroxamine was discovered in 1963 and was granted an FDA approval in 1968. Novartis began its production in the brand name of Despiral. So this has to be injected subcutaneously for a long period of time. So this is not orally active, and you must remember that this has to be injected subcutaneously. Another 2 iron chelators, both synthetic, namely deferisirox and deferiprone, were granted FDA approval in 2005 and 2011, merely after 40 years of despiroxamine approval. So these itself tell the story that how difficult it is to design a human compatible iron chelator. Both the synthetic compounds are orally active and are produced as tablets, and these are available in market. So we'll quickly review the comparative features of these 3 iron chelators: despiroxamine, deferisirox, and deferiprone. These are the clinically approved, FDA approved chelators. And we'll compare their this chelation therapy with phlebotomy, another method of attenuating iron overload. So despiroxamine is a hexadentate chelator, whereas deferisirox and deferiprone are tridentate and bidentate. So despiroxamine is not absorbed in gut, so it is not orally active, and so it is given as an injection, administered almost daily for 8 to 24 hours, leading to very low patient compatibility. On the other hand, deferricidox and deferiprone both are orally bioavailable and are given as tablets. However, even with those advantages, deferricidox and deferiprone are not being used so much in clinical studies because of their very high adverse effects, especially in myelosuppressions. Despiroxamine, on the other hand, has limited side effects, so very little side effects, mostly being local rashes over this injection spot, and which can be dealt locally. But another thing is that deferricidox treatment is very costly. If you can see the price, it is of much higher than the other costs, the cost of other things. So deferricidox treatment is very costly, and so it is not feasible for many third world countries. Another feature we must remember that despiroxamine bounds to iron in a hexadentate fashion, leaving no room for iron to be redox active because it is tightly bound. On the other hand, chelators like deferricidox, deferiprone, which bind iron in a tridentate and bidentate fashion, leaves a room for iron to be redox active because if it finds a another because it can be free from this bidentate or tridentate. So doctors and researchers rely on defruxamine, that is DFO, more than the other two for chelation treatment. And DFO has been used for chelation treatment for last almost 60 years. However, as we have seen, all these chelators have some limitations. For example, despiroxamine lacks in oral bioavailability, so uh, it is given as an injection, and also uh, it cannot penetrate the cells. So if it cannot go inside the cells, so it cannot chelate the intracellular labile iron pool. So the intracellular labile iron pool is untouched by despiroxamine. Another thing is that the half-life of despiroxamine is also very low, which means that the frequency of dosing will be very high. In fact, despiroxamine is injected almost every day, as we have seen earlier. The other two chelators, deferricidox, deferiprone, as I have said earlier, leads to quite severe side effects. This has been highlighted in some media reports. As you can see in my slide, there are some media reports where this has been highlighted and also has been in the center of some serious controversies. So the need of the hour is to develop more efficient chelators and also more efficient therapy protocols. To start with some of the advances that has been done towards these approved iron chelators in a more efficient way, a unique therapy with a combination of despiroxamine and deferiprone was tried. Despiroxamine being a very good chelator of blood plasma iron, but it cannot go inside the cells. So it was combined with deferiprone, which can go inside the cells. So a combination of chelators, one which can chelate the labile plasma iron and another which can chelate the labile iron pool, were combined. So in the extracellular compartment, the iron-bound deferricidox will transfer the iron to the free despiroxamine. So this deferiprone goes inside the cells uh, to bind the labile iron pool, then come outside, then uh, donates this iron to despiroxamine, and this iron-bound despiroxamine is secreted out of the body. And this deferiprone again goes into inside the cells to bind that labile iron pool. The non-transferrin bound iron in the blood serum will also be chelated by despiroxamine and will be thrown out of the body through renal or feces route. This particular method of combining the two iron chelators have shown remarkable effect in attenuating iron overload. How do I know? How do we know that? Because it is done basically by measuring the serum ferritin. Because we have seen that serum ferritin is a marker for iron overload. So in almost all the studies, the serum ferritin level was found to be decreasing after each therapy. As we have discussed earlier, Serum ferritin level is a marker of iron overload. So this particular therapy has been a very successful method in attenuating iron overload and has been in phase 2 clinical trial now. Apart from using two different clinically approved chelators, there is also a need of improved iron chelator. As you all understand, improvement can be done either by discovery of new chelator or by modification of existing approved iron chelator. For a new chelator to be effective, some aspects are needed to be taken care. For example, its selectivity and affinity towards iron should be very high. The metal complex should be very stable. Chelator should have the proper balance of lipophilicity and hydrophilicity go inside the cells. Above less toxic to normal pass all the categories of absorption, distribution, metabolism, and so there are some chelator which passed all these studies in clinical trial. Ferroxamine, as we have already discussed, is already clinically approved. This is a cedarophore. So I have kept the structure here again to show that this another cedarophore, that is desperithiazine, was discovered in 1980, and it was orally active, but it showed nephrotoxicity, and so there were no clinical application. But this was the backbone of many of the chelators which are now in clinical trial. So one of them is deferritin. As you can derivative of this deferritin. This is in 1999. It was not nephrotoxic, so it entered in 2007, and now phase 2 clinical trial is going on. So now another derivative, deferritrazole, it was discovered in 2007. It was not, but it was neurotoxic. So it failed in clinical trial. So another molecule, deferritrazole, that also failed in the clinic. That also failed in the clinical trial. But another derivative, that is SP420, was synthesized in 2010 and been taken using several hydroxypyridones, several hydroxyquinone, albomycin, etc. And we hope that one of these will get clinical approval. Option of we must remember Ferroxamine is the chelator of choice, because people are using this Ferroxamine and almost every knowledge of this Ferroxamine. So it is better, or it is advised by desperoxamine to gain some to Apart from the chelation of labile iron pool in different organs of human body, an ideal chelator should also have the property to chelate excess iron in brain. As you remember in the earlier slide, I have shown you that iron is very essential for brain because because brain is one of the most uh, important part of our body and it needs iron for its metabolism. So brain is a developing organ and so the requirement of iron in brain is quite high in young days. In case of iron overload, brain is also flooded with excess iron and is stored in different portions of the brain. Iron can start neurodegeneration leading to different diseases like Alzheimer's disease, Parkinson disease, etc. This is an MRI of a brain of an iron overloaded patient. Position of iron in several parts. So, what is special about the brain? Like every other organ, a cell-permeable chelator should be able to chelate excess iron from brain. But this is not the case. The brain is protected by a tight junction of endothelial cells called as blood-brain barrier, which does not allow entry to the unwanted compounds. Desperoxamine is not blood-brain barrier permeable, so it cannot enter brain. Uh, Although in one study, desperoxamine was shown to be neuroprotective, but that is quite controversial. But other chelators, the ferricidox, chloroquine, morphine, etc., are blood-brain barrier permeable, although their efficacies are questionable and still under study. So, to have a blood-brain barrier permeable desperoxamine conjugate, synthesize different blood-brain barrier permeable cell-penetrating peptides. And two of them, penetratin and TAT, were conjugated to desperoxamine. And their efficacies were examined in vitro. These conjugates were able to reduce both labile plasma iron in blood and also the intracellular labile iron pool. Most importantly, these conjugates were able to take out the excess iron from kidney, it will be very nice and very important from a medical point of view. What people did is prepared this kind of desperoxamine nanoparticle conjugate, and after a single dose of intravenous injection, the nanochelator showed favorable pharmacokinetic properties such as kidney-specific biodistribution and rapid renal excretion. Desperoxamine. dose, second subcutaneous administration of nanochelators improved pharmacokinetics, dynamics, and the increase in efficiency of urinary iron excretion was much more increased. And thirdly, daily subcutaneous injections of the nanochelators for 5 days for iron overload uh, decreased iron levels in serum also and liver. Further, the nanochelate significantly reduced kidney damage caused by iron overload without any nephrotoxicity. That is the most important, that it showed no nephrotoxicity at all. So this renal chelatable nanochelate provides enhanced efficiency and safety. So in conclusion, all the strategies so discussed appear very promising, although investigations are largely in preclinical stage. Each strategy has its own advantage, but in-depth study is pending. In these circumstances, there is a huge scope left in the field, particularly when around 2% of the total population in the world is treated for iron overload. In 2018-19, there was a news in India, or is actually truth, that a lot of thalassemia patients were in danger when Novartis suddenly reduced the production of despiroxamine due to some reasons, technical reasons. So this incident was an eye opener for all those who are associated and actually reiterated the importance of an ideal iron chelator. Cannot see any solution in the near future, but the work is in progress and the major breakthrough can be there anytime. So, I must acknowledge Professor Breno Esposito, who was my postdoc guide in Brazil, and he actually introduced me to this subject, and my colleagues at Bioorganic Division, and Samir, my good friend, and other organizers for this for giving me an opportunity to present this work. So, thank you for listening these things on a Sunday. Thanks.
Thank you very much, Dr. Goswami, to enrich us with very nice atom chemistry and its role and mechanism of action in our body, how we can get rid of that. And of course, from your suggestion, we should not be only vegetarian but also non-veg also. So
Yeah.
it's a
Yeah.
open to all to ask question to our. I can get from the chat box one question from Preeti Karmakar. As per the list, it was shown girls absorb iron milligram per day is more than boys. Then why most of women suffer from anemia in comparison to that of men?
Uh, it is not generalized that women are more vulnerable to anemia, but that is in because mostly because of our social things, that women are less what should I say, that they are less they take this food as less important thing from their childhood, and so the build up of iron levels in their body is much more
chat box. So I think we will not extend the session. So with this question and answer session, I, on behalf of Department of Chemistry, Saldiha College, must thank Dr. Kosami for his nice presentation and sharing his important time. Thank you once again. We extend our heartfelt gratitude to you, Dr. Kosami. Now, our last speaker is our youngest friend, Dr. Ramprasad Mishra from Germany, scientific researcher, Humboldt University of Berlin. We will talk on recent studies on thermal stability of protein, example of retinal protein. Dr. Mishra is a scientific researcher at the Humboldt University of Berlin, Germany. Earlier to this position, he was a postdoctoral fellow in the Weizmann Institute of Science, Israel. He did his doctoral studies in the Indian Association for the Cultivation of Science in Kolkata. His research interest encompasses biophysical chemistry, physical organic chemistry, and molecular statistics. Dr. Mishra was an integrated PhD fellow of ISS, visiting postgraduate research student at the University of Warwick, UK, and postdoctoral fellow at the University of Pittsburgh, USA. Till date, he has authored and co-authored 21 research articles in peer-reviewed journals and 2 books. He is a member of Royal Society of Chemistry. With this brief introduction, I would like to request Dr. Mishra to take the control of the screen for his presentation. Please, Dr. Mishra. Yes, go ahead. Extra 9. Extra student 9. Extra student 9. University student গুলো এখন 9.
We also know that actually protein is present in our body and they function numerous roles. Therefore, they are essential for human life. So to give some examples, that we know that the retinal proteins assist us in dim light vision. Antibodies, they bind to foreign particles such as bacteria and viruses and prevent us from diseases borne by them. And also enzymes that are there, those are also proteins. They carry out thousands of chemical reactions in our cells in the body. Also, the proteins are rich source of essential amino acids, which actually regulate immune functions as well as help in our body in building muscles. And so the second question arises that why studies of thermal stability of the protein are very important. So this is important due to many reasons. So out of them, I can mention only two, that these many thermally stable proteins are useful for industrial applications. And actually, the thermal stability of the protein is very important from us from many diseases. So if we just mention one of the very important study on protein thermal stability, we can know that this protein thermal stability issue is actually a multi-scale in nature. That means we can study at the molecular level. At the molecular level means the interaction among the amino acids and the protein level. And as we know that the proteins in our body does not stay as actually interact with several other biomolecules in our body. So we can also study this protein thermal stability at the structural level. So one of the important thing aspect is to mention that also we can study the thermal stability of a protein at different levels, but they are not actually very isolated. They sometimes are very related to each other. So to give an example, I can mention the story of the muscles of Atlantic salmon fish. So we know that at cold temperatures, when we put our hand in a cold water, what happens? Our muscles becomes rigid. We cannot function in the cold. But the salmon fish that actually stays in Atlantic Ocean, that temperature is very low, like below 10 degree centigrade. But they still function properly in that even cold temperature. So this can be mentioned that one animal is fine at cold temperature while others are not. So this thing can be very understood at the molecular level of protein. So here, one of the recent studies this year shown that a single amino acid substitution in salmon this tropomyosin, the protein that is present in the tail muscle of the fish, actually required flexibility in cold water. So to explain detail, what happens in this that in version of the topomyosin, by circular dichroism spectroscopic studies, which I think is not very important to show here. So we understand from different experiments that the retinal bound retinal is very important activity of thermophilic rod of cells. So here, okay, we have done another experiment like from the other way, that if the hydrolysis of the retinal protonated sheet base is important in thermal denature, if we prepare a PR actually film in which there is no bulk water, like we put the protein in a quartz plate and allow it to dry out the water, and finally we put it in a high vacuum, so no bulk water finally remains there. So we have found that if we compare the circular dichroism spectroscopy in the UV region, which is around 190 to 300, we can see that even at 100 degrees centigrade, there is no change in the protein secondary structure. So we know that at even 90 degrees centigrade in presence of water, the protein denature, but when there is no water present in the vicinity of the protein, that means in dry film, the protein can survive more than 100 degree centigrade. So what we did, we took out the protein film again and solubilize in water.
to have an idea about thermal properties of protein molecules with good example of retinal collagen. We enriched a lot from your lecture. Now it is open to all to ask question to our speaker. So I think there is one question from chat box from Dr. Kartik Gupta. What is the chemistry of causing retinitis pigmentosa leading to night blindness in some people? Is there any treatment for this disease till date? So, is there any question from our dear participant? I can find in the chat box. So, with this question and answer session, I, on behalf of Department of Chemistry, Saldiha College, must thank Dr. Mishra for his nice presentation and sharing his ideas and thoughts with us. Thank you once again. We extend our heartfelt gratitude to you. With this lecture, yes, yes, thank you. With this lecture, we are at the end of the technical session. Now, may I request Dr. Rana, the organizing secretary of the webinar, for vote of hands of the and close the session.